Hi, I'm Millie, and I'm going to be talking to you about investigating the effects of Culicoides saliva on phagocytosis in bovine monocytes. So first, a little bit of background. So I'm working within the orbivirus research group on blue tongue virus. So blue tongue is an orbivirus within the Reoviridae family. It's a double-stranded RNA genome with 10 linear segments, and it causes blue tongue disease in ruminants. And the symptoms vary depending on the host. So sheep, which are the most affected, display symptoms such as mouth ulcers, drooling, swelling of the mouth, head and neck, inflammation of the coronary band, fever and lameness. And this picture here is a picture of quite badly infected sheep. Cattle, on the other hand, remain mostly asymptomatic, but they're an important reservoir of the disease and are therefore very important to study in regards to the control of the disease. So the virus is transmitted by Culicoides midges. So a bit of preliminary data, off of which my study was based, was taken from my supervisor's thesis, so Dr. Lindsay Cook, where she infected bovine peripheral blood mononuclear cells, so PBMCs, with an MOI of 0.1 in the presence or absence of saliva. She then harvested these cells at 0, 24, 48 and 72 hours post-infection and internally labelled them for blue tongue viral structural protein, VSP, and non-structural protein, NS2. So VSP shows for viral infection and NS2 shows replication within the cells. And she gated on a CD14 positive population, so these were monocytes. So as you can see here on the top graph, this shows the percentage of CD14 positive cells that express blue tongue VSP. And so the darker bar is the cells that were infected with blue tongue alone, and the lighter bar are the cells infected with blue tongue and saliva. And as you can see, over time there is a gradual increase in infection when incubated with saliva at the time of infection. And this difference increases over time, so at 24 hours compared to 72 hours there is a greater difference. And then on the second graph, so this is looking at NS2, which shows viral replication within these cells. And as you can see here, the trend is the same. So there is a great increase between the cells that are treated with saliva. They become, the virus replicates in these cells more than in cells infected with blue tongue alone. So what we know from this? We know that Culicoides saliva increases blue tongue infection and replication in bovine monocytes. We know that this effect is only seen when saliva is added at the time of infection. So in her thesis, Lindsay also looked at the effect of adding the saliva at different time points. So when she added it at 6 hours post-infection, the effect was still visible, but much reduced. And when the saliva was added at 24 hours post-infection, there was then no difference between the infection and replication rates of the virus. So this suggests that the saliva is having its effect on the early stages of viral infection, and possibly on viral entry. And we know that blue tongue can use several entry pathways, including macropinocytosis and clefairy-mediated endocytosis, for example. And we know that monocytes, so the cells of interest here, are antigen-presenting cells that use phagocytosis. So my research. I am looking at the effect of Culicoides saliva on early blue tongue virus infection in primary ruminant cells, and specifically monocytes. And for this, I'm investigating two main areas. So the effects of Culicoides saliva on uptake pathways in bovine monocytes, and the effect of Culicoides saliva on early time points of blue tongue infection, specifically between 0 and 24 hours post-infection. And today, in the interest of time, I'm going to be focusing on the first one, so the effects of Culicoides saliva on uptake pathways in these cells, and specifically on phagocytosis. So my hypothesis was that Culicoides saliva increases phagocytosis in bovine monocytes, and for this I used fluorescent E. coli beads, which are currently used to study the entry pathway, and I assessed the uptake of these beads by flow cytometry. So this is just a quick overview of my methods. So first I isolated bovine PBMCs from blood. I then incubated them for an hour at room temperature with or without saliva. I then treated them with the E. coli beads, with or without saliva again, for 15 minutes. I then labelled monocytes with a fluorescent CD14 marker and looked at the uptake of the beads within this population by flow cytometry. So first, I looked at the effect of adding 1 microgram per ml saliva to these cells, and I included a 4-degree plate as a control, as biological processes should be reduced at this temperature. So the first graph shows the percentage of CD14 cells, the percentage of uptake in these cells, so the percentage of cells taking up the particles and this was standardised to account for animal-to-animal variation. So as you can see, there is an increase when treated with saliva at 4 degrees, and this difference is also seen at 37 degrees. There was, however, no difference observed between the two temperatures. And then the second graph, which is the mean fluorescent intensity of these particles. And so the MFI looks at more on a cell-to-cell basis. So the higher the MFI, the more each cell is taking up, rather than the percentage of cells, which obviously looks at it at a population level. So basically, the higher the MFI, the more each cell is taking up. And as you can see here, the trend is similar. So there is a higher MFI when treated with saliva, both at 4 degrees and at 37 degrees, and there is no significant difference once again between the two temperatures. So this suggests that the Culicoides saliva increases phagocytosis in bovine monocytes. So next I wanted to look at the effect of varying concentrations of saliva. And for this I used 0, 0.01, 0.1 and 1 microgram per ml of saliva. And I repeated this with 2 different animals. And so in the interest of this one, since there was only 2 replicates, I didn't want to normalise the data. Instead I'm showing you that despite there being variation between the 2 animals, the trend was consistent within them both. So the circles represent cow number 1, and the squares represent cow number 2, and the trend is upwards with increasing concentrations. And this can be seen at both 4 degrees and at 37 degrees, and is also the case when looking at MFI. So this suggests that the increase in E. coli uptake is dependent on saliva concentration. So next I wanted to look at LPS. So in Lindsay's thesis, she found that LPS also increased blue tongue infection and replication, and this is using the purified LPS. The reason that she looked into this was because LPS was found to be present in Culicoides saliva. So I wanted to see whether or not this effect was also observed in when looking at E. coli uptake and therefore phagocytosis. So the first graph again shows the percentage of cells taking up these particles. And as you can see, there is a difference again observed between E. coli only and E. coli and saliva. However, there was no difference observed when cells were treated with LPS compared to the untreated cells. And this is the case both at 4 and at 37 degrees. This is also the case when looking at MFI, as you can see in the second graph. And this just suggests that the LPS, so the purified LPS, isn't having the same effect when it comes to the E. coli uptake. So in conclusion, we found that saliva increases E. coli particles in bovine increases the uptake of E. coli particles in bovine monocytes. That this effect doesn't seem to be temperature dependent. That the increase in E. coli uptake is dependent on the concentration of saliva used. And that this effect is not observed when E. coli is treated with purified LPS. So what's next? It would be good to use saliva with LPS removed, just to confirm that the LPS is not having this effect. Maybe the LPS is responsible but needs to interact with other components of the saliva. It would also be good to use phagocytosis inhibitors during an infection experiment to determine if blue tongue enters bovine monocytes via phagocytosis and if this is the only pathway that is used by these cells. And then it would be good to repeat these experiments using bovine monocytes, using the different entry pathways as well, to see whether or not so pathways such as macropanocytosis and clathrin-mediated endocytosis are affected. I would like to thank the Ilbivirus Research Group and my supervisors at the RVC as well for their help throughout this project. Thank you very much for listening. Do you have any questions?